Hello, I didn't see you come in. Welcome to the tutorial of your protein graphing assignment. Here, we're going to be analyzing data for Biology 105 involving the protein assay, looking at the effectiveness of different buffers for lysing E. coli cells. So the metric we're using for assessing effectiveness of those buffers is how much protein gets freed up when we expose the E. coli to the different buffers. All right, let's go ahead and just get started. If you're looking at my lab section, I have a data set available for you on Canvas. I've highlighted it right here. You shouldn't have any trouble finding it. We're going to go ahead and look at that next to the lab handout and explain what's going on. All right, so we've got here what your data output should look like, okay? Essentially, each one of these cells represents one of the wells in the 96 well plate that you loaded. If you look at this loading scheme on the right, it's showing us that these three columns are all replicates, and these three columns are all replicates. Okay? We'll have to reference the protocol a little bit to know exactly what's going on. But let's start by organizing our data a little better. Okay. So, these three cells right here, they all represent an analysis of tube A. These three represent tube B, tube C, etc. So let's go ahead and look at what tube A, B, C, etc. are. They are this series of doubling dilutions, okay? Tube A contains a 2 milligram per milliliter stock solution. Let's go ahead and make a little header here in the Excel file. We're going to go with protein concentration. And I'm actually going to shorthand that by putting the word protein here into square brackets indicating concentration. Now our units are going to be in micrograms per milliliter. Alrighty, very cool. Let's resize that column so we have enough room to really see what's going on. Okay, so the first row, the concentration is 2,000 micrograms per milliliter. Then it's down to 1,000 because that's a 1/2 dilution, 100 microliters from tube A plus 100 microliters of diluent, and so on. Okay, each one of these is just 1/2 the value of the cell above. I'm going to make Excel do that work for me. And then the last cell, it only contains diluent, so its protein concentration is 0. Fantastic. Okay, now let's go ahead. I'm actually going to click column E. I'm going to right click and I'm going to hit insert because I want to have space for my average values. And let's do that again because we're going to need more data labels. Okay. All right. So this column here is going to be average values. And we're going to make Excel take those average values. So what you need to do is an Excel formula or function. You're going to type equals symbol and then type the word average and then open parentheses, so Shift 9, all right, and then select the values that you want Excel to take the average of and hit enter. Now, if you want to double check that Excel is taking the average of the right values, click the cell and then click the formula bar at the top. And it'll show you that what you have set up here is an Excel function where that cell is taking the 3 cells to the left and averaging their values. What does that mean? That means if I grab the lower right of this cell and drag it down, it's going to do that exact same function for every single one of these cells, but it's going to transpose the cells that it's referencing, all right? Or it's going to continue to reference the 3 cells to the left of the cell that you've typed in. So now we've got protein concentrations and we've got average absorbance values. And right there, that's everything we need to generate a standard curve. So let's go ahead and do it really quickly. Now, I'm going to show you a little Excel pro tip, all right? Just select this, click and drag to select these values, the protein concentrations. Then hold the Control key, All right. If you are using a Windows, I believe it's the Command key if you're using a Mac operating system. So hold that Control and then click and drag these values for average absorbance. All right. Then go to the top and hit Insert. You want the scatter plot right here, and drop a scatter plot in there. All right. Stupendous. So now you have all those values, okay, graphed. What else do we need to do? Let's go ahead and just full screen Excel for a moment so we can zoom in and get a better view of this stuff. First things first, okay, let's get rid of those grid lines and make the standard curve look a little cluttered. Now what do we need? We need to add in a trend line. So go to Chart Elements right here at the upper right. If you're using a Mac, you'll have to go up to Chart Design at the top and then go over to Add Chart Elements at the top left and put the trend line in here. The functions are the same, okay? You're also going to need to go More Trend Line Options. It's going to open up this little window on the right. You then need to make sure that you're clicking this bar symbol right here, okay? Now, hidden by my face is a checkbox that says Display Equation on Chart. Let's go ahead and move my face over to the opposite side for a little bit. There I go. I'm on the other side. I should be on the other side now. Fantastic. Hit Display Equation on Chart, and boom. There we go. Now you can see the standard curve equation for our line. And remember here, y equals absorbance, x equals concentration. So now we can plug in absorbance values for those unknown wells that we did with the rest of the plate, okay? And we can solve for concentrations. I'm going to drag that thing down a little bit, put it over there. Okay, now let's go ahead and get back to our data and reorganizing it a little bit. Zooming out. Here we go. So if we scroll down to our loading scheme, you'll see that columns 4, 5, and 6, okay? These three values are replicates of tube 1. These three values are replicates of tube 1, but diluted. These three values are replicates of tube 2, and so on and so forth. So what are those tubes? All right, what do those numbers represent? Well, they represent different buffers. Buffer 1 is hypotonic, 2 is isotonic, and 3 is hypertonic, okay, with 4 being an isotonic solution plus detergent designed to break open those cells with chemical reactions rather than with tonicity. All right, let's go ahead and make another header right there in column F for buffer, okay? So we've got buffer, this is hypotonic, okay, as is the buffer below, that one's just diluted. Then we've got isotonic, as is the cell below, that one's just diluted. Then we have hypertonic, and you see what I'm doing here. We're just going to go ahead and wrap that up. And then this last one is isotonic. I'm just going to put the plus symbol in, detergent. That works. That's our labeling scheme. Another fun little Excel pro tip, if I click the F column and highlight it right here, and then I go to where I can resize that manually, or you can just double click right there, and it'll resize it to fit all the text that you have in that column, which is nice when you're referencing data. Okay, cool. So we have the buffers. Now what are we missing? We are missing the dilution, okay? So remember, tube 1D is the hypotonic buffer, but it's been diluted. This is useful because what if one of these concentrations is outside the range of our standard curve? We do the dilutions too to sort of cover our bases and also to give us two values that we can compare. So we're going to go ahead. I'm going to click the column G here, and I'm going to right click. I'm going to insert, and I'm going to add in another section for dilution. Okay, and I'm going to actually make these the dilution factor. The dilution factor is the number in the denominator of a dilution that has 1 in the numerator. All right, that's at least how we're going to talk about it. So what I mean is when we do 1/5 dilutions, the dilution factor is 5. That'll make our math easier in a second. Now that column's a little large, so I'm going to resize it with that same trick I used before. So in the undiluted tubes, the dilution factor is 1. The other tubes were diluted by adding 20 microliters of tube 1 to 80 microliters of diluent, which is a 1/5 dilution, making the dilution factor 5. So we're just going to 1, 5, 1, 5, 1, and 5. And there we go. Now we have our dilution factors all right there. Now this is basically all the information we need to answer our big question. So I'm going to zoom into the Excel sheet of which one of these buffers was the most effective at lysine E. coli cells. All right. So first thing I'm going to do is I'm going to take the average of these values for absorbance. Boom. Same function, type it in, click it, drag it. There we go. We're done. All right. But now I need to convert these values into protein concentration. All right. So I'm just going to make a header for protein. All right. Now immediately I'm seeing some problems here. Look at the highest absorbance value from our standard curve indicated right here. It's 1.64253. That means that any values higher than that, we cannot trust for use with our standard curve, which means that these two values here are outside of our range, okay? We'll still calculate the protein concentrations, but keep in mind, we're not going to be able to trust those values. So I'm just going to highlight those cells so that I don't accidentally discuss them or use that data. All right, let's go ahead and rearrange this equation, our standard curve equation, to solve for x. So go to this first cell right here, put equals, okay? Now, you probably went over this in lab, but you got to remember to throw parentheses in at the beginning here for the order of operations. You'll see what I mean. So we're going to tell this equals, I'm going to actually, 1 second here. We're going to actually zoom in so you can see the text a little better. Okay. And I'm going to drag the, boom, boom, boom. I'm going to drag our standard curve equation underneath so you can see exactly what I'm doing. Okay, so the standard curve equation I need is I want to solve y minus, all right, y being absorbance value, minus 0.1687, but that needs to be in parentheses, and then divide that by 0.0008, all right, to solve for x. Now, I'm going to tell Excel, with the parentheses, right, that my y value is the cell directly to the left of wherever I'm typing this formula, because that's my absorbance value right there, okay? Subtract 0.01687, close the parentheses, and then divide it by 0.0008, all right? And sure enough, that protein concentration value is also outside the range of our standard curve. Now, if you've been tracking what I've been doing so far pretty well, you're probably thinking already that, oh, if I just grab that cell and drag the lower right corner all the way down, it's going to solve for the standard curve for all those absorbance values, solve for x. And you are correct. All right? So there it is. It's now been converted into protein concentrations. We're still missing something, though, okay? We want the undiluted concentrations, okay? So I'm just going to make a header that says undiluted, and we need to incorporate that dilution factor, all right? So once again, I'm sorry, I've actually highlighted all these cells. I'm going to undo that. All right, we're going to go with a no fill real quick. And then we're going to highlight these two cells once again as not useful values, okay, because they're too high. All right, so now what we need to do is we need to actually take the calculated protein concentrations and multiply them by the dilution factors. It's not that bad. Check this out. I'm going to put equals this protein concentration and then times, which is the asterisk symbol, so shift 8, the dilution factor over here, and hit enter. Now it's going to be the same for this first row because that was not diluted. When we grab this cell and drag it this time, now we're calculating the undiluted protein concentrations. All right. Now you're going to see that some of these values are actually higher than our standard curve, but that's okay because the values that we actually measured over here were not outside of our standard curve. We did the same thing with the carbohydrates in the previous lab, right? Same thing. We dilute the sample and then measure and then calculate what the concentration was before the dilution. So we still can't use these two values. Do not use these two values. They're too high, all right? But the rest of this can all be compared. So you're going to have some questions here about which were the most effective at lysing cells and freeing up proteins. Alrighty. Now, I don't want to do the final steps here for you, but keep in mind that you could compare, that you can take the average of some of these values. Okay? And you can rearrange this data as you'd like. Alrighty. So, for example, you could take the average of these two values, and that's a fair representation of what's in buffer 2. And the same is true of these two values right here. You could take the average, you could take the average of these two, which I've done here, to get a representation of what's in buffer 3. But remember, you cannot incorporate those values where the concentration was too high. So you're going to need to figure out with a data set like this, or with this specific data set, if you're working in my lab, just exactly which buffer is the most effective at freeing up proteins or lysing cells. So why do we do it this way? Okay, think about this. We were able to conduct the standard curve and measure our samples all in the same 96 well plate, which means that we did the whole experiment in the same way. We had a lot more control and we had a lot more assurance that the experimental groups and the standard curve groups, okay, were all handled in the same way. Anyway, I hope this recording has been useful. I hope you enjoyed it. And thank you for watching. I have been and will continue to be Jonathan Montgomery. Goodbye.